thank you so much for being here. This is our, I think, first after Corona in-person R&D day. Today focusing on multiple sclerosis and our Vidofludimus calcium program. It's really nice that we can be here in person in San Francisco. Thank you so much for coming. And also welcome to those who listen to our webcast. Not moving. Click, yeah. Usually I read the long forward-looking statements, but today I just show them. You know that we have forward-looking statements in the presentation. So as I said, we will focus on the multiple sclerosis program today. We will walk a little bit about, through the unmet medical need in MS, and then dive into our Vidofludimus calcium program, the mode of action, and then also the ongoing and completed clinical trials in RRMS, RMS, and PMS. And then Daniel will share some insights on our IP portfolio, and then look into the future a little bit on upcoming milestones and what can we expect in the next months and years. And then in the end, we will talk a little bit about positioning and the commercial potential. And then in the end, very important, we will have outside a networking lunch. So the Hotel Nikko team will prepare this here. Please feel free to stay another couple of minutes and have some lunch with us. Maybe one word on asking questions. We don't have a dedicated Q&A session today. Just whenever you have a question, just throw it in throughout the presentation. Our colleagues will be happy to answer them. Good. Maybe quick introductions. I forgot to introduce myself, of course. So I'm Jessica. I think everybody knows me. I'm VP Investor Relations Communications at Immunic. Then presenting today, we will have Dr. Daniel Vitt, our CEO and President, Dr. Hella Kohlhof, our CSO, and Dr. Andreas Muehler, our CMO. And then I'm also very, very happy that Dr. Zuming Sun is here today. He's a professor at the Department of Molecular Imaging and Therapy at City of Hope in Duarte. Maybe Hella can say some words about him and his research in your section. But if you have any research-related questions related to NOR1, etc., it's great to have Zuming here today who can share some insights. And with this, I will hand over to Daniel to kick this off.
Good. Thank you, Jessica. Yeah, once again, welcome also from my side. And when we prepared that meeting, so we had a discussion on what is the right way to make sure everybody understands that the treatment landscape of multiple sclerosis is totally changing. So there are a lot of things going on, and on the research side, from the clinical experts, you see a lot of papers about understanding the disease. What can we do today with the treatments which are available? What can't be done with the current treatments? And luckily, Immunic has developed a molecule, or is developing a molecule, Vidofludimus calcium, which has properties which could change in principle, in generally, the way we treat multiple sclerosis. And this is a big message. And it's honestly, we think that 50% of the MS problem is not solved today. So it's a big challenge for research and for industry to offer better solutions. And therefore, we want to talk about it. And therefore, we dedicated this R&D day to multiple sclerosis and the understanding the unmet medical need. And then Andreas will start with a little bit more diving into the details of what can be done today and what's missing. My colleague Hella then will introduce the target NOR1 we are working on, where we believe NOR1 is one of the first real-life promises to address neuroprotection and to prevent neural death in a focal inflammation independent way. And then we will try to set that in the context of where the industry is right now. And that is all in the context of today we have April. So in 1 year, we will read out our progressive MS study, the second one on this slide here, the CALLIPER study, which is designed to answer some of those questions you will see today. And Andreas will also, in his clinical section, remind you that we already got positive interim data from the CALLIPER study in the second half of last year, which was super exciting for us, and I think also for our collaborators and KOLs in that space. We also continue our execution of our phase 3 program, the ENSURE studies in relapsing MS. But I think the research focus is a little bit more on the neuroprotective side today, because we think that's a very unique new approach. And to put that in a context, if you go back a couple of decades ago, MS, there was no treatment at all. It all started with the interferons, and I think it was a tough job for people developing a treatment in a market where people said there is no market because there is no treatment. So the first wave were just injectables, so interferons, and also Copaxone and so forth. And then we call it the second wave, were more targeted immune suppressive, including some oral drugs we have right now available. And then there should be the third wave, which is so far an attempt on goal, but there's nothing approved, which is directed to neuroprotection and the so-called relapse-independent disability worsening, or PIRA. And before I hand over to Andreas to talk about the unmet medical need and also introduce the scale, EDSS score, and further ways to measure the progression of multiple sclerosis, I want to draw your attention to why this is a totally unmet medical need and what is the problem. So the elephant in the room is disability worsening. If you look on that slide here, we try to really plot the two important parameters of disability worsening and the two main contributions. One is relapse-associated worsening, which we can treat with today's treatment in a pretty nice way for the cost of safety and tolerability issues. On the other hand, this is the so-called PIRA, so progression independent of relapse activity. And that is more or less not addressed with the current treatments. And I think we all believe that the introduction of the anti-CD20 therapy was a big step for patients. And we need to continue on that way. And Ocrevus is approved for primary progressive MS, which I think was an important step for patients there as well. But what's next? What's coming to fill the space where you can address both RAW and PIRA in the same way? And with that, Andreas, I would like to hand over to you to talk in the first section here about the unmet medical need in MS.
Thank you, Daniel. Good morning, everybody. I wanted to start here, talk a little bit about the unmet medical need in MS. And as you all know, this is a disease of about 1 million patients in the US, more prevalent in women than in men. It starts relatively early. So these patients, when they get the diagnosis for them, the question then is, how do I look in 20 or 30 years? Can I live independently? Can I function? So this disability question is the main question that patients have on their mind. And we measure this as EDSS score, usually. I will talk about this a little bit today. That there are other ways, other sides of disability that actually are important to patients as well, that we sometimes don't measure with EDSS. But EDSS is kind of what we usually use for measuring disability in patients in clinical trials and outside clinical trials. And this EDSS score is something, it's a progressive disease, so it's always getting a little bit worse over time. So patients start here on the left side at diagnosis often, and then basically slowly but surely move to higher scores of disability, which of course is making their life, their personal life, their social life, their professional life a little bit more difficult. So if you ask patients, what is your biggest concern, is not whether they have 2 or 3 relapses in the next 30 years. Their biggest concern is, can I live independently in 20 or 30 years? Or how do I live? And how do I manage my life in 20 or 30 years? So it's a disease that starts early in life. And that's why you have that as a patient, often that very much this long-term view. So there are different forms of MS, of course. So this slide is a little bit as an introduction here for the different forms of MS we have. There's what the FDA definition is. It's kind of the upper row here. The FDA has a relapsing form of MS or relapsing MS, and then on the right side as progressive MS. The relapsing side and some of the progressive MS forms, they're basically different stages of the disease. So if you look at the first three basically quadrants there, they're basically the same disease, but at different stages in time often. So that's usually what happens to patients. So they often, 85% or so of patients starts with the relapsing-remitting form of MS on the most left quadrant here, where the disease is really dominated in terms of if it's in its appearance to patients and to neurologists by having MRI lesions and having relapse events. And in between, you have times when you don't have relapses, but you can see MRI lesions appearing. And that's really how most patients start. Later on, these patients, they're basically these intervals between relapses become longer and longer. And at some point, this leads to the stage which we call active secondary progressive MS or active SPMS, where you still have relapses, but more rare. You still have MRI lesions, but more rare. And there's longer intervals in between these relapses where you have an impairment or the decline of disability over time. And it becomes more and more apparent that you have this decline of disability in between relapses. And of course, later on, often these patients go into what's then called non-active SPMS, which is a little bit a wrong designation of the disease. Non-active disease doesn't mean that you no longer have disease or that's not longer active, but the most outward appearance of disease activity, relapses and MRI lesions, are no longer present. So you are deteriorating in disability, sometimes even faster than before, but you don't have relapses or MRI lesions. And then it's called non-active SPMS. So all of these three forms happen over time, and there's no clear-cut differentiation sometimes. There's a little overlap, little gray area, and you will see it sometimes in these clinical results where you have patients with non-active, for example, SPMS, that a few of them, if you do a clinical trial, will have some lesions and maybe even, sometimes rarely, they have a few relapses. But in principle, it's because this is kind of the transition between these three different stages of the same disease is sometimes difficult to really mark on a certain date. And on the right side, there is what's called primary progressive MS. This is a little bit a different form of MS in the sense that these patients start with something where disability is slowly worsening, even at diagnosis, and you don't really have, at least as textbook, you don't have relapses. Yeah, so this starts without relapses and goes right into the progressive phase. In real life, some of these progressive, primary progressive MS patients rarely have some relapses as well, which doesn't change the diagnosis. They really start with a disease right from the diagnosis that is mainly progressing and really doesn't have the relapsing part. So these are the four forms of MS. So, and the FDA considers the right two quadrants as progressive MS, the left two quadrants as relapsing MS or RMS. So that's when we talk about RMS and PMS, these are the forms of MS that we're talking about. So what has become apparent over the last 2 years only, with a publication in 2022 about a very large meta-analysis of how disability appears in MS patients in about almost 30,000 MS patients in clinical trials. And what was really new and really shocking for the medical MS community is that even in early relapsing-remitting disease, you basically already have a progressive disease, meaning that half of the disability worsening that you have in relapsing-remitting disease is already relapse independent. later on, I will talk about a little bit what that's PIRA. And so you have in principle, MS is purely a progressive disease that's just overlaid initially with some relapse activity in MRI lesions. And since they're so often in appearance, so dominant, if you have a relapse and the patient really deteriorates from one day to another with a lot of neurological deficits, we have too much emphasis on these relapse events and don't recognize that behind, even in the relapsing forms of MS, behind this is a progressive disease that goes on. And it was something that really changed the view of the medical community about MS. And it was really this, and different experts now call it differently, and they're still fighting in terms of what is the right name. So some call them invisible disability accumulation that happens in relapsing forms of MS. Or others say smoldering disease. But in principle, the technical term now is basically PIRA. There's a lot of PIRA already in the relapsing disease, progression independent of relapse activity. And it's about half of the disability, even in very early disease. So that is something that really changed everybody's view on MS. So, and the reason why this was able to be really so obvious now is that this meta-analysis that I talked about looked at these increases in disability that happened in these patients that they were observed in clinical trials, and looked whether this increase in disability, that's kind of the gray shaded area here, whether it was in correlation and temporal correlation with a relapse event, or whether it was away from a relapse event and basically happened without a relapse in between. So you can consider this a little bit, this raw, that's the upper row, which is the relapse-associated worsening, that you have a relapse, from one day to another, you basically get worse. You have neurological deficits. And usually, these patients recover within 30, 60, or 90 days and fully recover from that relapse. But some of the patients don't really recover fully. There's sequelae. And so you have a remaining neurological deficit or disability worsening, and that's called relapse-associated worsening. But there's also, like I said, about 50% of the disability worsenings, even in early disease and relapsing disease, is not associated with relapses. They happen in these intervals in between relapses. And that was really the surprise. And that's why I think this lower row, which is called PIRA, or progression independent of relapse activity, that is so dominant in early forms of MS and relapsing MS. And basically, it's the exclusive way of acquiring disability in the progressive forms of MS was really, I think, what has now put really the light fully on PIRA as the unmet medical need in MRI. And I tell you exactly why. In the same meta-analysis that was published 2 years ago, they looked at what is the difference between patients in these trials that receive placebo versus who receive these approved MS treatments that we have available now. And what you see here on the left side is patients who receive treatments, they have a lot of less relapses. Of course, this is how we approve treatments. Yes, you have less relapses versus placebo versus a comparator. But on the right side, that was kind of the interesting, really finding in this meta-analysis, that if you basically treat patients with a disease-modifying drug in these clinical trials, as compared to placebo, you have a lot less relapse-associated worsening. Yes, you decrease the number of relapses, but and therefore you also decrease the number of relapse-associated worsening, because they don't have these relapses from which they cannot fully recover. But in principle, the amount of PIRA, progression independent of relapse activity, stayed the same. What happens if you take away raw events but keep the PIRA events the same is that the amount of PIRA events in post-treatment with the currently available disease-modifying drugs is much higher. Yes, because you decrease one category of these relapse events, of these disability events, but you don't do anything to the other category, PIRA. So, and basically the conclusion of this meta-analysis was also that the currently available anti-inflammatory disease-modifying drugs MS do basically almost nothing to progression-independent relapse activity. So they work on disability as well, yes, but they work through decreasing relapse-associated worsening because they decrease relapses. So it also, since this time, the medical community has really said, well, if we really want to improve patients in the future, we have to address and decrease PIRA by treatments. And that's, I think, where the medical community is now. And that's, I think, where I think really the spotlight of the research in MS is. So this was kind of the unmet medical need in terms of the treatment and the efficacy part. There's also a safety part that's often overlooked in MS. Yes, because you have so many treatments available. And you say, well, they have so many treatments. They're happy to take these drugs and so forth. That there is also a safety component of the unmet medical need. And the reason is that these patients are often very early, when they're diagnosed, they're very early in their life. They're 25, 30, 35 years old. They have a family life, they have a professional life. And for them, being able to continue these daily activities is important. And the one thing that often is, you also have these patients, is that early on in their disease, they don't really have a huge disease burden yet. So they don't know their course of MS and they're unsure, but they look at these drugs, and most of these drugs that are available now have some sort of safety or monitoring burden that comes for these patients. And when you think about the different drugs that are available now, there is differences in, of course, in efficacy, yes, especially, for example, between an anti-CD20 drug like ocrelizumab and a very early drug like glatiramer acetate, that is one of the early drugs in MS, really doesn't have that much efficacy as compared to anti-CD20 drugs. And you see on the right side here, if you look at the number of patients, not the market in terms of dollars, but you look at the number of patients, they're almost the same number of patients still on glatiramer acetate than on anti-CD20 drugs. That's amazing. You would say, why would a patient choose glatiramer acetate? There is a large proportion of patients who really are willing to do a compromise between their safety and what they require from the treatment and the efficacy of the drug. And that's why glatiramer acetate is still so much used by a larger proportion or by a sizable segment of the MS patients. And that was basically on the left side. There was good studies out there. There are several, I'm just focusing here on one, that really show that there's a sizable patient segment that says safety is as important to me, especially early in disease, because I have a life and I look at these risks of these drugs also and basically choose how I choose my treatment. So there is an opening also for a drug that has a robust efficacy, but has a much more acceptable safety and tolerability profile to this sizable segment of patients. And we believe that Vidofludimus calcium, and I will show you some of the data later on, the safety data, really addresses very well this segment of patients for whom the safety is as important as efficacy. And we have, and I'll show you more of the data, but here's just a slide that summarizes that in all of the risk and tolerability categories in which MS drugs have some shortcomings, basically we have shown that our safety profile is indistinguishable from placebo in the trials that we have done so far in MS. So we have now an exposure of more than 1,800 patients and human subjects with Vidofludimus calcium, and we have not seen a signal for hepatotoxicity or an increase in liver enzyme elevation rates as compared to placebo. We have not seen a signal of increased infections as compared to placebo. So there's, at this point, I think we believe this is a very unique safety profile for vidofludimus calcium. And so if we talk about vidofludimus calcium, we believe we're the blue balloon who stands out in a crowd of different disease-modifying treatments for two reasons. We're the first drug, really, we believe, that has a neuroprotective effect. And we'll talk about this in a second, the Nurr1 mechanism that really addresses or may address the PIRA issue that's completely unaddressed by anti-inflammatory drugs. And the second is that we have a safety and tolerability profile that is very acceptable to those patients which value safety and tolerability in the same way as they value efficacy. So that's kind of why we believe this is really a drug that addresses both sides of these unmet medical need in MS. And I'll stop here because I was asked to take questions before we go into the next section.
Maybe I can go first. Do we have PIRA data from some other trials that, by Asian that maybe help contextualize? I know you showed the meta-analysis. I'm just curious if that PIRA, how that, numerically looks like.
That's an interesting question. So the question was whether we have other data other than the meta-analysis on PIRA. Not much yet. And the reason is that this concept of PIRA and RAW was really established by this meta-analysis. And it required so many patients put together in a meta-analysis. But I can almost assure you that every trial that you see coming out in the future will have that disability worsening characterized as either PIRA or RAW. And you see this now with trials ongoing that they'll do this. And also the definition of what we consider RAW and PIRA came from this meta-analysis. There was never an agreement before how to do this. And everybody refers now to that paper. So Fred Lublin and his paper is really probably very well referenced in the last 2 years in MS, including by us. You're welcome. Well, if no further question, then I would hand over to my colleague Hella to talk about the mechanism of action of vidofludimus calcium, especially I think the newer information that we have on our Nurr1 mechanism of action, that has neuroprotective potential for this drug. Thank you.
Thank you. Okay, so let's go on with the mode of action of vidofludimus calcium. As Andreas said already, vidofludimus calcium addresses the smoldering neurodegeneration. And actually, when we started first, vidofludimus calcium was a pure DH/ODH inhibitor, very selective, very safe. And that was already great. But then we figured out from the clinical data, actually, that there must be something more. There must be something on preventing neurodegeneration. And we digged a bit deeper and figured out, wow, vidofludimus calcium is a potent Nurr1 activator. And Nurr1 is a nuclear receptor. It is known already from Parkinson's disease that it has neuroprotective functions, and there are coming more and more publications for multiple sclerosis that Nurr1 really plays an important role here for preventing neurodegeneration. So with having vidofludimus calcium, we really think that we have the first-in-class Nurr1 activator, and that it really can target the improvement of physical and mental ability of multiple sclerosis patients. So vidofludimus calcium is a Nurr1 activator. It protects neurons from cell death, and there's a continuous effect which is independent of focal inflammation. That's a Nurr1 activity here. On the other hand, as I said, we know that, or it was shown already, that vidofludimus calcium is a potent DH/ODH inhibitor. And this is twofold interesting, actually, because we have this selective anti-inflammatory effect which reduces focal inflammation. And I will come to this later in the presentation. And in addition to this, it was known already that DH/ODH inhibition has an intrinsic antiviral effect. And then we figured out that this prevents as well the reactivation of Epstein-Barr virus and could stop cross-reactive immune responses. So it was a, how is it called? Is it called a hat trick if you have a triple attack here? I don't know what the right word is, but this was very intriguing to us. So coming to Nurr1. Nurr1 as a nuclear receptor, which is involved in neuroprotection. And it can be quite complex in the brain. Actually, we have a lot of different cells interacting with each other. So it's not only neurons which express Nurr1, but also microglia and astrocytes. And just to show you here in a very simplified overview, we see the expression of Nurr1 in microglia, which are the cells like macrophages, brain resident macrophages. There Nurr1 is expressed, and you can activate microglia, and then they can produce pro-inflammatory cytokines, having a negative impact on the survival of neurons, for example. In addition to this, astrocytes express Nurr1 as well. And astrocytes are important for clearance of, if you have too many neurotransmitters, it's important for supporting the blood-brain barrier and so on. And of course, Nurr1 is expressed in the neurons as well. And just to simplify it here, we put it together as Nurr1 activation mediates neuronal survival. Then Nurr1 activation prevents microglia and astrocyte-driven neurotoxicity in the brain. And Nurr1 activation in motor neurons may halt the neurodegeneration and disability progression. And I will come to more details later. So when we looked first for Vidofludimus calcium as a Nurr1 activator, it was shown in different ways. So first of all, we tested the compound in different reporter assay systems. So the GAL4 system, and then we looked as well on full-length Nurr1 reporter systems. And what you see here is a very strong activation of the Nurr1 signal here. So 3-fold or even up to 5.5-fold activation. So this is really a very strong activator. And there were some publications on simvastatin, for example, or other, well, more or less potent Nurr1 activators, which are not selective actually. And there it was always just up to maybe 2-fold activation. So here you see that Vidofludimus calcium is a really potent and strong activator of Nurr1 signaling. In addition to this, we looked as well for direct binding. We used ITC here, you see it on the right, and we're able to show that Vidofludimus calcium is indeed a direct binder of Nurr1, and then it activates Nurr1. Coming again to the different cells which express Nurr1 and what is happening in there, we put it here again with the microglia and astrocytes. If you activate Nurr1 in these cells, you have a reduction of pro-inflammatory cytokines, of nitrogen oxide, and of reactive oxygen species, which have a negative impact on neuronal survival. And then when you activate Nurr1 in neurons directly, you reduce oxidative stress and you have better neuronal survival and differentiation. What was shown in Parkinson patients or in neurons from Parkinson's disease patients was that it increases dopaminergic neurotransmission, and in addition, it has a positive impact on myelination. So to summarize it, Nurr1 activation is believed to be involved in halting neurodegeneration and disability progression. And of course, we did some experiments, or better say, Su-Ming's lab with Hong Min did these experiments. So if you have questions and if I cannot answer them, we can ask Su-Ming. That's perfect that he's here. So first of all, Vidofludimus calcium was tested for its Nurr1 activation signals in microglia. So microglia were stimulated with LPS, and then they produced interferon gamma, IL-6, and TNF-alpha, which are all pro-inflammatory cytokines. You see it here in the graphs in human microglia cells, HMC3 cells, that first, with the first column here, DMSO was set to 1. When you do the stimulation with LPS, you see that you have an increase of IL-6 production. And when you then add immuates rate of Vidofludimus calcium, which is here the name 1260, that's a lab name, then you see a reduction down to the unstimulated level or even below here. So we were able to show that with Vidofludimus calcium, indeed, we can prevent the activation of microglia producing interferon gamma, IL-6, and TNF-alpha. So having, again, an indirect via Nurr1 activation anti-inflammatory response here. In addition to this, we looked for direct target genes. That was more or less indirect, but we looked as well for direct target genes in human microglia cells and in neurons as well. So you see here on the left, the expression of the primary target gene VMAT, what is vesicular monoamine transporter 2, important for dopaminergic neurons, but also expressed as a direct target of Nurr1 in astrocytes and in microglia. What you see here is on the left that you have a wonderful dose-dependent upregulation of VMAT2 in the microglia. And you see that already with 30 nanomolar, you have more than twofold expression of VMAT2 in the cell system. Coming to the next effect, so looking for direct effects on Nurr1 in neurons. We looked for this in the so-called SHSY5Y cells. It's a human neuronal cell line, and as well in the murine neuronal cell line. And here it is known that Nurr1 activation can prevent apoptosis of neuronal cells if you induce apoptosis by combination of TNF-alpha and cycloheximide. So the test system was set as follows, that first the cells were treated for 1 hour with a compound, then the DAS challenge with TNF-alpha and cycloheximide was added to the cell culture, and then 6 hours later, the apoptosis analysis was done. And here again, you wonderfully see that with having Vidofludimus calcium inducing Nurr1 activation, it's really possible to prevent or to ameliorate the apoptosis induction that was triggered by TNF-alpha and cycloheximide. So Vidofludimus calcium prevents or at least ameliorates, dependent on the cell system, the apoptosis induction in neural cells via the Nurr1 activation. Then in the next step, an animal model was performed, an EAE model, so a typical model for RMS in mice. And in this model, Su-Ming's group was able to show that Vidofludimus calcium is first of all active in the EAE model, and we used 2 different concentrations or 2 different doses here, 30 and 150 mg/kg. And then we looked for gene expression of the target and of a primary target gene. So the target NR4A2 is Nurr1, and you see that in the brain of the mice, so really in the tissue of interest, you see already an upregulation of the target. There was, in a similar way, there was an upregulation of the target via 838 or Vidofludimus calcium treatment in the spinal cord. What you see as well is that the expression of Nurr1 in the spinal cord seems to be lower compared to the brain. So we see target expression induction via Vidofludimus calcium. And then what is even more important is when we looked for primary target gene, which is in this case tyrosine hydroxylase, we were able to show that we have a dose-dependent induction of tyrosine hydroxylase. So really hitting Nurr1 in the brain, which then translates into upregulation of tyrosine hydroxylase, a primary target gene in the tissue of interest. Next to this, we were interested in BDNF, brain-derived neurotrophic factor, which is a target of Nurr1 as well. And for this, we looked in the heart plasma and were able to show, yes, indeed, we see a dose-dependent upregulation of the expression of mature, which is neuroprotective again, of mature BDNF in the treated mice suffering from EAE. So that was the path for Nurr1 and the direct neuroprotection here. As I said in the beginning, Vidofludimus calcium is a potent and selective DHODH inhibitor, and there are some very good features of this. This is, for example, that inhibiting DHODH really targets only the highly metabolically activated immune cells. In this case, we looked for high producers of IL-17F, IL-17A, and interferon gamma high producer cells. And it is known that hyperactive or high affinity cells are specifically dependent on DHODH inhibition because they have a high metabolic turnover in these cells. And the point is why we think that it's really happening like this is if you have a high affinity cell, you have a high production of for example, IL-17 or interferon gamma. And for this, you need high amounts of mRNA. So it's not just blocking the proliferation of these high affinity cells. It's really blocking the production of these high amounts of cytokines like IL-17A, F, or interferon gamma. And what you see here on the FACS plot on the right-hand side is that when you see the circles there, that compared from vehicle to Vidofludimus calcium, you really see that only the high-producing cells for IL-17F and interferon gamma are reduced. There are remaining IL-17F and interferon gamma cells, but they have much lower expressions of IL-17 and interferon gamma because this is just like the covering the normal expression of cytokines, what you always need, actually. And yeah, the next point with DHODH, it's an intrinsic inhibitor, or, well, it provides broad spectrum antiviral activity against different viruses, and we tested it in many different viruses. Of course, we tested it against SARS-CoV-2. We looked for HCV, human CMV, HIV, and arenavirus like Lassa fever virus, and we're always able to show that Vidofludimus calcium is a potent inhibitor of infection or has a potent antiviral activity in these infected cells. How does it work? So actually viruses rely on the host cell's infrastructure for their replication. And if you inhibit DHODH by Vidofludimus calcium, for example, this leads to a depletion of pyrimidine nucleotides. And these are needed again for the production of viral RNA and DNA, so for the virus genome. And of course, this is needed for the production of viral proteins via mRNA. And we looked as well for Epstein-Barr virus because this is so prominent in the root cause of EBV as well. So we looked as well for Epstein-Barr virus replication and reactivation. And this means that by targeting the host cell metabolism, Vidofludimus calcium has shown to be active against different RNA and DNA viruses in vitro, including a strong anti-EBV activity. So coming to EBV, there were some really nice publications, and it started, I think, 3 years ago, that it was shown that Epstein-Barr virus is essential for the onset of multiple sclerosis and is involved in ongoing autoimmunity. And it was shown that there's a clear association between EBV infection and occurrence of MS. There's a 32-fold higher increased risk in EBV-infected patients. And then the cause or the reason why it has these activity or these outcome in multiple sclerosis was that cross-reactive antibodies were identified. And these antibodies are cross-reactive between Epstein-Barr virus, antigen EBNA1, and the CNS protein glial-chem. And that was found in the CSF of MS patients. So it means that EBV infection and then the reactivation, because I think 95% of everybody here is infected with Epstein-Barr virus, but the reactivation seems to be an ongoing trigger for the immune system in multiple sclerosis patients. And yeah, as I said, there was some really nice publication, and then it was even shown that multiple sclerosis is not only preceded by the EBV infection, but also associated with a broader EBV-specific T-cell receptor repertoire. So counting for an ongoing reactivation in the MS patients. As I said, we tested Vidofludimus calcium for its ability to prevent reactivation of Epstein-Barr virus. That was tested in different cell types, like in the Akata cells, all these more or less B cells, because it's a reservoir for Epstein-Barr virus. And on the left, you can see a dose-dependent inhibition of the reactivation of Epstein-Barr virus, and that was shown as well in Raji and B95.8 cells, where the lytic reactivation of Epstein-Barr virus is strongly reduced by Vidofludimus calcium. What does it mean for MS patients? So the prevention of EBV reactivation by Vidofludimus calcium could provide a long-term benefit for MS patients. When you see on the left, this is the infection cycle. So first we have epithelial cells which are infected, and then you have the viral entry and the replication. This enters the B cells. If you have activated B cells, they provide the antigen to T cells. Normally, in a healthy person, you just attack Epstein-Barr virus and it goes to the latent cycle. Everything is fine. If you don't have a problem with your immune system, it will stay there for, well, all the time, actually. So in patients where you have a diminished immune system or not known triggers from the outside, you can have a reactivation of Epstein-Barr virus. And then again, you have a shedding virus, so you have the lytic cycle and shedding new viruses. With Vidofludimus calcium, actually, we can inhibit, in principle, the EBV replication and, what is much more important, the reactivation of Epstein-Barr virus. And what does it mean for the MS patients now? So in principle, coming to the cross-reactive antibodies, it means that with each reactivation and infection cycle, you have a newly generated humoral immune response, interesting word, immune response, which bears the risk of newly generated cross-reactive antibodies. And actually, this is a process that is called somatic hypermutation, and it really happens in the brain of patients as well. So with having a blockade of the recurrent reactivation cycle of Epstein-Barr virus by Vidofludimus calcium, this might therefore then provide a long-term benefit because you reduce the constant neurodestructive trigger of Epstein-Barr virus. And that was, interestingly, we were able to show the antiviral activity of Vidofludimus calcium in an MS trial that was not intended in the beginning, but actually the recruitment of this cohort was during pandemics. So the patients were, of course, tested for SARS-CoV-2 infection or COVID-19. And what you see here on the left is first, during this trial in the 10 milligram cohort, so very low dose for MS patients, there was already a reduction of any infection seen in these patients compared to placebo. And then we looked deeper into the relation with COVID-19, and we're able to show that indeed, if you treat the patients with just 10 milligrams of iminoatrate or Vidofludimus calcium, you have a reduction of COVID-19 infections here down to 8.5%. And at the same time, we looked for the development of antibodies against SARS-CoV-2, and we're able to show that the treatment with Vidofludimus calcium does not interfere with the antibody development against SARS-CoV-2 or during the SARS-CoV-2 infection. So what does it mean? Where's the connection between SARS-CoV-2, Epstein-Barr virus, MS, post-COVID symptoms? And this could be actually fatigue. And it is known that fatigue is the most common symptom in post-COVID syndrome patients and as well in MS patients. And it was claimed that Epstein-Barr virus reactivation is seen in both MS patients and in PCS patients, but not in the healthy controls. So EBV infection and the reactivation is known to induce the strong fatigue in some patients, whereas others have mild symptoms, but there is a connection. And with Vidofludimus calcium, we block the reactivation of Epstein-Barr virus, and we were able to, and we reduce fatigue. We were able to show to reduce fatigue in the COVID-1 study. So in the COVID study we did in the past. So it means that by preventing the reactivation of Epstein-Barr virus, Vidofludimus calcium might reduce fatigue in MS patients as well. And this will be tested in the phase 3 ENSURE studies as well. And Andreas will talk a bit more in detail about this. Okay, coming to the summary here. That's the same slide. Just to remind you, we have with Vidofludimus calcium a potent Nurr1 activator, which protects neurons from cell death. And so we have this additional wonderful effect here, which will be shown in the clinical studies. Let's get fingers crossed. We have shown it already partially, but we will show it in the ongoing clinical studies. And in addition to this, we have this potent DHODH inhibition, which is absolutely helpful for selective anti-inflammatory effects, which reduce the focal inflammation. And we have the antiviral effect, which prevents EBV reactivation and the harm from Epstein-Barr virus in this. So that was on the mode of action. Are there questions? Okay, that was the, great.
In terms of the DHODH, essentially is blocking something that's
Proliferate.
cells, yes. Are there any potential effects on, say, you can see on lymphocytes that are also proliferating due to demyelinating events?
It is, we haven't checked for oligodendrocytes, but what is important is that DHODH inhibition does not firstly affect fast proliferating cells. It's really the high producing cells which are important. Because if cells proliferate, they need double of the DNA. But if they are high producers for IL-17 or interferon gamma, they need up to 100-fold more nucleotides, which need to be produced by the de novo synthesis pyrimidine synthesis. So it's, well, I would say the effect is much more on hyperactivated cells than on just normally proliferating cells. And we looked as well in an animal model where we checked exactly this for neutrophils and dosed the animals up to, I think, 300 mg/kg, so really high doses here. And there was no impact on white blood cells, which are known to proliferate quite quickly. And so it's really a selective effect on high producing and highly metabolically active cells. Any further questions? Yeah, sure.
You know, you sort of laid out the underlying fear. I'm just curious how neuroprotective may really, it targeting fear may really set up for that cross.
So it could be that Vidofludimus calcium Nurr1 activation is of course not only targeting the neuronal survival, but it targets as well the neuroinflammation. So it could be that in addition to the DHODH inhibition, that this is of course why we have this wonderful NFL reduction. So coming from both sides with Nurr1 activation and with DHODH inhibition. Thank you.
All right. Oops, I wanted to now come to our clinical trial results and go through the different the trials here. I wanted to start really with, I think, the most amazing data we had in 2023 in October that we published the interim results of our CALLIPER trial. This is the phase 2 trial in progressive MS patients. And here I wanted to introduce you to the trial first. It's a phase 2 trial. It's actually a fairly large phase 2 trial that looks at a dose of 45 milligrams of vidofludimus calcium versus placebo in patients with progressive disease. We do serial MRI in these patients because the primary endpoint is brain atrophy, or the what the scientific name is kind of the annualized rate of a brain volume change. And we did also, in parallel to all the other assessments of biomarkers, so neurofilament and GFAP in these patients. So we highlighted here the week 24 because we did the interim analysis if a certain number of patients then had a neurofilament and GFAP data at week 24. So the study is fully enrolled since last year. It's a phase 2 study with 467 patients enrolled. It includes progressive MS patients of different diagnosis or different types of progressive disease. It contains about 1/3 of the study is primary progressive MS. So that's the far right quadrant, if you remember some of the first slides. And 2/3 of the trial is non-active secondary progressive MS patients. That was kind of the third quadrant. Yeah? So these are the two types of progressive MS. And they look a little bit different in terms of their baseline characteristics from relapsing disease, relapsing MS, because they're usually older. The average age was over 50 years. And also their EDSS, so their disability, is already quite progressed. The average EDSS score of 5.5. So that's already quite advanced disease. We looked in this interim analysis at biomarker. So neurofilament, of course, is very well known in a lot of neurodegenerative diseases, and it's also very well known in relapsing MS. But there is increasing evidence that's a very useful biomarker also for progressive disease. Neurofilament is a peptide that originates when neurons, and especially the axonal proteins, are degraded. So during the degradation, these neurofilament peptides first appear in CSF, and then also you can measure them in blood. And so it was now shown that these NFL elevations, increases, precede confirmed disability events, and also that there is a time between certain disability events and basically having neurofilament in the blood of progressive MS patients. So it's a useful marker in progressive MS. So the main study that showed us the usefulness of neurofilament in a progressive MS population was the Oratorio study. Oratorio study is the phase 3 study of ocrelizumab, an anti-CD20 therapy, in primary progressive MS patients. And this was just released last year, middle of last year. And it was a long-term analysis of the disability worsening and the correlation to neurofilament in these patients. So what the authors did in this analysis is they took and re-baselined the NFL data 48 weeks after the start of the therapy with ocrelizumab. So the idea is the third infusion of ocrelizumab. The idea is that at that point, the anti-inflammatory disease is effectively decreased by ocrelizumab, by 3 infusions of ocrelizumab. And then you re-baseline NFL, and you basically say, now I'm looking at patients who have higher NFL values despite having no focal inflammatory disease, and have lower NFL values despite having no focal inflammatory disease. And you then focus over 8 years. What happens to these patients with low and high NFL? And it basically showed very clearly that those who have high NFL and have no focal inflammatory disease have a higher risk of developing confirmed disability worsening than those with lower NFL. So patients with low NFL have really a lower future risk of disability. So that was very nicely shown in this very long-term study, 8-year study. So this is the same oratorio study, but it shows you the change in neurofilament early on in the trial. And you saw that, and you see here that the treatment, ocrelizumab, decreases neurofilament very effectively and basically early on in this trial as compared to placebo. And at week 24, it's the same time point that we have reported our data in its interim analysis, you have about a 12.4% decrease in neurofilament as compared to placebo for ocrelizumab at week 24. So that's just as a marker in terms of what you could see as an amplitude of neurofilament changes as compared to placebo in these progressive MS patients. So we had a pre-planned, that was in the protocol, a pre-planned interim analysis. The study remained blinded, so the individual treatment assignments of patients remained blinded because we didn't want to unblind the study. However, we got group level data for these biomarkers. So the biomarkers were basically neurofilament and also another emerging biomarker, GFAP. The interim analysis included 203 patients at the normal size of a phase 2 study, of 203 MS patients, which had both baseline and week 24 data in these biomarkers. At that time was also, by the way, a unblinded safety analysis done by the IDMC, which showed no new safety alert and recommended to basically continue this trial without any changes. So this was good. So this slide summarizes the main results in terms of the biomarkers here. So we looked at the change of neurofilament between baseline and week 24 for all of the patients. Now in all of the 203 patients. So if you're on placebo treatment, and this is a progressive disease, your neurofilament increases over time. It actually goes up. And then with the treatment, neurofilament actually goes down. So you have about a 22% differential between those patients who are in active treatment versus placebo up to week 24. And actually, this was shown to be statistically significant already in this interim analysis, in a post-hoc analysis. So on the left side, you see again the differential between active treatment and placebo, the 22.4% that I just realized, and then broken down for the different diagnoses, the different subtypes of progressive MS. So primary progressive, PPMS, non-active secondary progressive, these are the two parts of that make up progressive disease. And you see there is a very consistent decrease of neurofilament in all of the subtypes. It's not driven by a certain subtypes. And then you have patients who have active secondary progressive. There's very few patients in there, but it will always happen that some of these patients develop relapses or lesions, and they basically also show a decrease. But I think it's very nice to see that in both primary progressive and non-active SPMS, they really have the same amplitude. It's really very consistent in this interim analysis. So again, on the left side is the 22.4% amplitude between active and placebo in the overall population. But here's now on the right side, we show what happens if patients had activity, inflammatory activity in this trial. So in the middle, these middle two means active progressive MS and non-active progressive MS. Means they have no relapses, they have no increase in T2 lesion load, they have no new gadolinium lesion in the trial. So basically, they're completely silent if they're non-active progressive MS. You still have a very sizable effect of neurofilament on neurofilament. And the same is true if you look at patients with active and non-active MRI. So active means you have lesions, which by textbook you shouldn't have, but they still some have. And then you have no lesions, and still having no lesions, meaning you have no focal inflammation going on in these patients. You still have a decrease of neurofilament. And that's remarkable because a lot of the, like I said, the anti-inflammatory treatments that are approved now and available in MS, they really only can do something about the active patients that have focal inflammatory disease because they're anti-inflammatory drugs. And that's basically here summarized on this slide. So on the left is basically the neurofilament, the amplitude of change between active and placebo in our interim analysis versus the oratorio trial, the 24-week data from the oratorio trial that I also shown you. And it basically shows that we're in the same amplitude of effect, or at least in the same amplitude of effect than ocrelizumab at week 24 in terms of differential between active and placebo treatment. But here in the middle, it's a very interesting study. It was a study, it's called EXPAND study. It was a phase 3 study in secondary progressive MS patients. It was done some time ago, about 10 years ago. And it was done with siponimod versus placebo. Siponimod is an S1P modulator. It's a purely anti-inflammatory drug. It sequesters lymphocytes and basically out of the blood, and basically by that not allowing them to go into the brain. So it's a purely anti-inflammatory mechanism. And they looked at active versus non-active SPMS. So active SPMS, you still have inflammation ongoing, you still actually have relapses. And you see, and that's on the right side of these four in the middle, they still have a very sizable effect on neurofilament, 21.4% differential between active treatment and placebo. This is actually at 1 year. That was the first time point where they had NFL data. And it shows that if you have focal inflammation ongoing, you have an anti-inflammatory drug, of course you have an effect on neurofilament. And of course, vidofludimus has an effect on neurofilament in these patients as well. But if you take siponimod, a purely anti-inflammatory drug, and that's kind of here in the middle, there's minus 4.4%, you basically have no effect on neurofilament in this population that doesn't have inflammation. But we still have a very sizable 20% effect with vidofludimus calcium, despite these patients have absolutely no focal inflammation left. So this is really telling for us, these four columns in the middle, that vidofludimus calcium does not behave like a purely anti-inflammatory drug. It actually works in a population where there's no focal inflammation left, and still has a sizable effect on neurofilament in this patient population. That's the most significant evidence of a clinical neuroprotective effect that we currently can present to you. And we hope that, of course, like Daniel said, in a year and 12 months from now, we have the full readout and we can show you more of this. And I think that's really these type of data that really made us very excited about this interim analysis. So overall, the interim analysis really showed that the effect of vidofludimus on neurofilament, on these biomarkers, goes beyond the known effect that anti-inflammatory drugs have. We think that also starts to indicate that this could be something that really goes at PIRA, goes at the high unmet medical need in non-active MS, and also I think especially non-active secondary progressive, where there's no treatments available at the moment, and it's a sizable population of the MS patients. Again, we will read this out in exactly 12 months from now, in April 2025. And we think that if progressive MS patients, in principle, they only have PIRA, they don't have RAW, yes? So if we show an effect on disability in April 2025, on disability outcome, it also informs kind of the outcome in the ENSURE trials, where, again, this is relapsing MS, where 50% of the disability is actually PIRA. So we believe that there's also a read-through to relapsing MS phase 3 trials, then with the positive, hopefully positive data that we can show you in 12 months. So that was kind of the short, at least for me, it's a short summary of the interim data. Any questions that I should answer here? Otherwise, I will speak. Sure, please.
So here, these are all placebo-adjusted numbers.
Yes.
So your effect on active SPMS looks, bigger than what you saw in the RMS.
Yes.
So I'm just trying to understand, like, why that is. That because the baseline is, or the control arm, is declining more in the progressive MS than RMS?
No, I would say if you look at the ends here for active SPMS, because we were focusing in this trial on non-active SPMS. Yeah, so in a lot of ways, I think it's a problem of N. Yeah, so we have 20 patients for both placebo and active at this point. And of course, if you have a focal inflammatory component to these, which these patients still have, you have both not only Nurr1 effect, but you also have the DHODH inhibition effect. So you have basically two mechanisms of action working to lower neurofilament. But I would say, let's look when we have more data and more patients with active SPMS, what the magnitude is. I would caution a little bit to make conclusions about the magnitude of the effect here. And we have actually the data I'll show you in a second in the phase 2 study, relapsing MS, which I think showed that we have a very good sizable effect on neurofilament in these patients.
Could that also be related to dose?
That's also just in like 2 minutes, I'll show you the dose linear effects. No, a very good introduction. Thank you for the next section. So before I go back to that section, I wanted to talk about something that we usually don't talk about, which is the functional outcomes in MS studies that we do, and which are part of our ENSURE and CALLIPER program in relapsing and progressive MS. And I think it's something that we don't usually talk about, because everybody focuses on physical disability that we measure with EDSS. Yes, and since all of the trials, the primary endpoint that in progressive MS trials and the key secondary endpoints is always kind of confirmed disability worsening, which is purely based on EDSS. There's also a lot of focus now in the MS community to look at outcomes that are really have a disability component for the patient, but are not measured by EDSS. And the three largest one is fatigue, cognitive decline, and quality of life issues. And these are three very important outcomes for the MS patients that we have to improve to make their life better, but it's not measured with EDSS. And I will focus on fatigue and cognitive decline today a little bit and talk about why they're so important to MS patients. And it's often called silent disability changes, yes, because you don't measure them in a clinical trial with EDSS, but they're happening and they're very important. And actually, there's lots of examples now, lots of studies now that show actually patients demand that treatments are switched, for example, or neurologists want to switch treatment because they see these changes in cognition, for example, and consider this so important that actually it leads to a therapy change. So there's a lot of functional readouts that we do in clinical trials always. And on the left side, as I think this is very established functional outcome there outside EDSS, which is called the 25-foot walk. Basically, you have a distance and the patient has to walk this as fast as he or she can. And then the 9-hole peg test, which also is a coordination test. And that has been used for quite a while. What we also in our trials always are focusing on is cognition. And I will explain to you what it is, but it shows you also that cognition really is declining, deteriorating with the longer you have the MS disease. And really, I think the big elephant in the room is fatigue in MS, because that's really the most important symptom besides physical disability for MS patients. And I will focus on cognition and fatigue. So first, cognition. So cognition is basically something that's hard to explain usually in a setting what cognition really means, but it's really a very prevalent and debilitating symptoms for MS patients. And I will explain why. And the other thing that's interesting about cognition, I will show you this in the next slide, is it's mostly independent of relapse activity. So it happens kind of independent of relapse. It's something that goes on in the background that is not related to relapses. So why is cognition changes so important for MS patients? So first of all, there's what you say in a psychological literature in terms of slowed cognitive processing speed. So what it means is patients have a hard time, for example, speaking speaking to a certain topic. They get interrupted in their thinking. It's hard for them to shift from topic to topic, and also recognition, for example, of certain of certain shapes, forms, and so forth take longer, and they recognize that. There's also, it's also a psychological term, it's kind of impaired executive function. So it basically means if you have complex problems, it's very hard for them to kind of see all of the different components of it. And what often happens to them is they say, I'm lost in a maze, I'm feeling stuck here. And they also, if they have certain complex tasks, they can't pay attention to it. And there are lots of examples from patients that they say that that's really makes their work life, their professional life, very, very difficult. And of course, you have these also periods of memory decline that they recognize as compared to the longer the disease goes on. And this was a study done now and published that basically showed that cognitive decline that you have in these patients are there really in correlation or happening at the same time concurrent to physical disability. So if the physical disability gets worse, is also the cognition gets worse. And interesting enough, half of the cognition decline, when a patient talks about cognition decline, is completely independent of physical disability. It's something that happens independent. And if you look at the independent cognitive declines that are not related to relapse, of course, most of them what's called a paracognitive decline. Yes, they're independent of relapses. So it's very interesting that seems to be going on, cognitive decline seems to be going on in the background, independent of relapses. So it's something more that belongs to that background process of MS, of that slow decline that goes on. So cognition is probably very much a marker of that as well. The really most important symptom for MS patients is MS fatigue. That happens in almost every patient in MS. Almost every patient knows this. And it's not only a physical component, it's a mental component of MS, and usually it's both. And we call it fatigue because it's not just sleepiness or exhaustion or something. It really goes beyond this. People don't have, the will to start certain things because they don't have enough energy to do certain activities, which of course inhibits their social and professional life. And it often also goes along with anxiety or mood changes and so forth. So that's really debilitating for MS patients. The interesting thing about MS fatigue is there is no treatment available, but also some of the treatments that you have available for fatigue, and there are certain drugs that are labeled for other forms of fatigue, they really don't work very well in MS patients. Of course, they get prescribed, but they really don't work that well. Coming back a little bit in terms of we have more and more new data where the fatigue in MS is coming from. And I think Hella started to talk a little bit about EBV reactivation. We have these very interesting data that were published 2 years ago that shows that if you look at EBV-infected MS patients and EBV-infected control patients, the T-cell repertoire, so kind of I always said, I'm not that scientific, so I always say kind of the top 10 list of hits what these T-cells see. For MS patients, this includes always 2 or 3 of the EBV antigens. For the control patients that are also EBV positive, they don't have these EBV antigens in their top 10 list, these T cells. So somehow MS patients have these MS patients see a lot more recurrently always seeing these EBV antigens that a control group that's also EBV infected doesn't see. So interesting. So there's some constant reactivation of EBV ongoing in MS patients. That was truly remarkable. And we actually see this in clinical trials. And think about it a little bit, it's not the EBV antibodies that you really see with activation. It's EBV DNA that now you have in your blood or in your saliva. And in the clinical trial, so you see here, for example, on the right side, you see that in different trials, in MS trials, you see about roughly 20% of time points, if you measure EBV DNA in saliva, you see DNA in there. So 20% of time, patients have a sizable and confirmed EBV reactivation because they have EBV DNA. And then if you take a antiviral drug like teriflunomide, another DHODH inhibitor, this really is only happening at about 10% of the time point. So there's already shown that with a antiviral therapy, you can decrease the time points that show EBV reactivation. So that's interesting, yes? Now let's go to, because we wanted to we learn also something from post-COVID or long COVID. And here this is a study basically on EBV reactivation in post-COVID patients. And to the left, you see different antigens and the antibodies that you measure in blood of these different EBV antigens. They don't differ between patients who have long COVID and don't have long COVID. So here, there's also plus LC, minus LC, that it's long COVID fatigue. And the plus, of course, they have long COVID fatigue, and minus, they don't have long COVID fatigue. And the antibodies titers are not different. However, to the right, if you look at how much EBV DNA they have in their throat washing, which is similar thing to saliva, there's a huge difference. So the detectable EBV DNA in throat washings basically happens in 50% of the COVID fatigue patients, but only 20% of the non-long COVID fatigue patients. So really, there seems to be a connection between EBV reactivation and fatigue. And also there was another study that basically looked at what are the long COVID symptoms in patients that have confirmed EBV reactivations. Fatigue is the #1 symptoms in patients that have long COVID, that have EBV, confirmed EBV reactivation. So again, there seems to be more and more data that somehow the EBV reactivation happens in both post-COVID and also in MS to a sizable degree. And there seems to be a connection to fatigue in patients that have EBV reactivation. And this, I think, has the hypothesis that maybe that's the same in MS, that those patients who have EBV reactivation, that we know a lot of them have ongoing, the T cells see EBV on an ongoing basis. We have the clinical data, and I'll show you them, Hella mentioned them, basically where we looked at the post-COVID symptoms in our COVID trial for Vidofludimus calcium. And there was a questionnaire about what post-COVID symptoms you have. Those patients have active treatment with 45 milligrams Vidofludimus calcium had much less fatigue. They only have about 50% of fatigue, whereas in the placebo patient had about 80% fatigue. So it was really, I think, already showing that an antiviral drug really were able to lower the number of fatigue. This was basically just giving you a little insight that beyond EDSS, we're also in our trials, we really look at other functional outcomes in these MS patients. Sorry, it's a probably more complicated story, but I hope I made it as simple as I could.
Is there a way to enrich for EBV gap in your MS study?
So EBV patients, because it's a precondition to MS, they're more or less 100% EBV infected.
The ones which have more.
Yes, and then yes, that's a good question. However, it's basically what I showed you, the 20% of time points. So it's very likely when you look at these trials that the patient comes, let's say, every 12 weeks to visit, and you can look at the saliva or you can look at blood for EBV DNA. That at some point they have, the next point they don't have it, then for next 2 they don't have it. So it would be very difficult to kind of only look at patients who at baseline have EBV DNA and only put these patients into the trial. So what you have to do, and that's also what we're doing in our trials, we measure EBV DNA more or less on every visit or on many visits. And it's more or less a statistical problem that you really decrease the number of time points where you have EBV DNA detected. That's probably the only way to deal with it. So far, I think it has been the only way to deal with it.
You were really hard on the EBV hypothesis, and then
Yeah,
Not as, but I think what experiments you're running and the clinical data in phase 3 with symptoms and, that could, put a lot of light on the hypothesis.
yeah. So I don't think we want to claim that EBV reactivation maybe makes MS worse. That's our trials are probably needed to be designed differently. However, MS fatigue is a very important symptoms. A lot of unmet medical need there. If I go to any MS Congress, there's at least one section that's only devoted to fatigue. Yes, that's always the amazing thing when I go there. So it's a really important issue, and it's a potential differentiator for this drug as well, that you say, we actually have the data to show that we can influence MS fatigue. That's, I think, why we're doing this, rather than solving the biological, question about the role of EBV in MS in general. Good. Very quickly, I will do as quickly as possible. I know. Yes. So the phase 2 study emphasis trial that we had in relapsing-remitting. This is kind of the first study that we did in MS with this drug. It had basically different doses in there. It looked at basically 24 weeks of blinded treatment and measured, it was basically an MRI study as a primary endpoint. We showed that there's a very strong reduction in the overall number of MRI lesions as compared to placebo here, 76% reduction of overall lesions, and then a 78% reduction of gadolinium enhancing lesions, highly statistically significant. We've shown that the 76 and 78% reduction in lesions that we've seen is very competitive as compared to very similar drugs in a similar setting. So that I think that compares well. So this is, I think, where I said, let's wait for the data. We have a very dose linear decrease in neurofilament in these patients who have active disease, meaning they have inflammation. Yes, focal inflammation. So we have dose linear decrease, which also I think is different from the basically dose response curve that you saw for lesions, where it was plateauing after 30. It actually goes down. Yes, and that also I think is an indicator that if neuroprotective effects play a role, maybe that's the reason why you have a dose linear effect in neurofilament. We actually saw also some hints on EDSS as a group on a group level basis, that it was stable on a group level over the 24 weeks in treatment groups, but was slightly increasing placebo. But of course, the really endpoint that is important to regulators is confirmed disability worsening. So you have a baseline EDSS, and then at some point in your trial, your EDSS value go down by a preset value. So you have that increase. So that's your trigger event. But then you want to avoid that it's just a random event. You look at 3 months or 6 months later, whether that increase and at EDSS and disability is basically persisting. So that's called the confirmation event. And only then for regulators is a confirmed disability worsening. And that is the important endpoint. So we did actually do an interim analysis for the emphasis trial 2 years or 2.5 years ago, where we looked at confirmed disability worsening from our phase 2 trial. And we could show that here we have a decrease in the confirmed disability event as compared to placebo, which was a first really good indication that we have an effect on disability in the relapsing population as well. So we also looked at, in the open label study, how many patients, there's more or less a Kaplan-Meier analysis, how many patients remained disability worsening free in this 2-year analysis here. And it was most of the patients, very few, about 5 to 6% of patients had any disability worsening event. And that's relatively low as compared to other drugs over the same 2-year period in terms of confirmed disability event. So it's an inter-study comparison, so you have to be careful. However, I think both consistently for 12 weeks and for 24 weeks, which is this one, confirmed disability worsening, we are on the lower end of drugs in terms of the number of confirmed disability worsenings over 2 years. So very interesting. So in this trial also, emphasis trial, we really also saw a remarkable safety and tolerability profile. So we have in terms of, on the right side here, you see liver enzyme elevations in two different categories. There was no difference between actively treated patients with vidofludimus calcium and placebo patients. And you also see on the left side is what the regulators really looking at is what's called Hy's Law assessment. So you have a concurrent increase of total bilirubin and liver enzyme elevations, and that gets you in trouble as a drug developer. We have no such cases. So we have no cases of Hy's Law here in our database. This is the data that I think Hella confirmed a little bit, already mentioned, that we have in vivo preclinical data that shows that you can give as much of the vidofludimus calcium as you want in an animal model. There is no anti-proliferative effect in bone marrow. So you can't use this as an anti-proliferative drug. That's kind of shown in this slide. Whereas if you take cyclophosphamide, you definitely have an anti-proliferative effect. Yes, that's the control here on the right side of each column. So you can suppress the immune cells. And we have also seen this clinically. We have no effect on hematology, no neutropenia, no lymphopenia, very stable hematology results in our trials. So, which also leads then, of course, to that you have no increase in infections. Yes, because you maintain the immunocompetence of these cells, and you have no increase in infection as compared to placebo. That's not normal for an immunomodulatory drug, but it's because it's a clean DHODH inhibitor. There's no increase in renal events as compared to placebo. And also in terms of the discontinuation rates in our phase 2 study, very low in active treatment, actually much lower than placebo treatment. It shows, first of all, it's safe and well tolerated for these patients, and they feel well treated in this active treatment. So very remarkable results. And I think that safety tolerability profile here is in comparison to, in different categories, to labeled drugs that are available on the market that all have some sort of safety or monitoring liability. And here, so far in all of these categories that are known for other MS drugs, we really haven't seen any signal. So we are quite happy about the safety tolerability profile of vidofludimus calcium. Any questions so far? Then
Can you just remind us why you chose the 30-minute dose? Also, I mean, your CDW data was better for the higher dose. I guess that goes well with CALLIPER, but
so the CDW data basically were not different for different doses. They were basically combining all of the doses. So, so the 30 milligram was based on that you have a plateauing effect after 30 milligrams for the anti-inflammatory effect, which are the most important thing for relapsing MS. Yes, but we took the 45 milligrams dose because there was this dose linear effect in neurofilament that basically says that you have an increasing effect of neurofilament that is different from the plateauing effect in MRI lesions.
the safety data was combined.
Yes, but there's also, do you see, there's no dose linear effect in safety. Yes. Just combining to have a little higher ends, yes, because otherwise, one event can create a whole. Yeah. Absolutely, especially now with other drugs in development in MS, having seen these Hy's Law cases, and that's absolutely on focus for the FDA. Yes. Good. I'll continue this very quickly. So we have an ongoing phase 3 program. You probably know that for relapsing MS. These are two basically identical twin studies, ENSURE 1 and ENSURE 2. They're done in about 1,050 patients each, placebo-controlled. There's a primary endpoint, of course, that's relapse-based for each trial. And then there's a combined key secondary endpoint that's only done in the pooled data set for confirmed disability worsening, which I think requires more patients to be statistically significant. And that's how the program was set up, also with regulators, to allow us this pooled analysis of disability worsening. There's a main treatment period of, a blinded treatment period of 72 weeks, which is kind of little bit between a year and a year and a half. We have basically powered the study here for a hazard ratio of 0.67 between these active treatment and placebo that led us to that patient population of about 1,050 patients per study. We have an interim analysis coming up this year for the ENSURE trials. This is a pre-planned interim analysis. It allows for a monitoring of the sample size and also for the relapse rates. And also it includes, it's likely not a very data-rich interim analysis, not as much as the CALLIPER trial. And since it's more or less an analysis that can only be done by an unblinded IDMC, and we only get recommendations. So we really, I think we don't expect that these are data that in the same sense as the interim analysis for CALLIPER. So this will happen in third quarter this year. So then just to reiterate, I think we're very much interested in fatigue and EBV reactivations because that's very much a potential differentiating factor for this drug. So we do measure fatigue with different questionnaires in our trials. And we look at the changes in EBV antibodies in blood, but also we look at EBV DNA in saliva in all of our trials. Good.
Andreas, there's one question from our digital audience here on ENSURE. What do you think is the bar for success?
The bar for what?
Success.
For
The
what?
bar for success.
The bar for success for the interim analysis.
No, for ENSURE overall.
But of course, meeting the primary endpoint and meeting the key secondary endpoint, that would be great because that's how the study is powered. And that also would allow us to speak to the FDA without any hesitation. Yes, I think that's I think the success. I think there's of course always different shades of success, but I think that's kind of the most important issue for success in our ENSURE trials.
It's a second order question.
Yeah. Yes.
The control arm that you have is a non-active. I'm sure they were because there are trials which use an active control arm. So I guess not having that makes it a very easy win, if you may. But I was also curious if you think about this sample size reassessment for later this year. What are the different scenarios, right? Like in terms of, I know you'll only get a binary answer, but like, have you planned, like, is it like X number of patients you have to increase the study sample size, or, there's a particular secondary endpoint you're, statistically trying to hit, and they are gonna, like,
No.
or is it just based on purely on that primary endpoint?
So it's really difficult to make any assessment without being unblinded. So that's why it's in the hands of the IDMC to give us recommendations. We ask them certain questions, but in principle, there could be very different recommendations that they give us. But I think the most likely outcome is they tell us to continue as planned. Yes, that's what we hope for, and that's the success for this interim analysis. But as with any interim analysis, I can't make predictions without having unblinded data. Yes, and of course, I will not be unblinded.
And you said how much follow-up? Like this is the whole 72-week follow-up in all patients.
So yes. So every patient is in the trial in the double-blind treatment period for 72 weeks. But the interim analysis, it basically takes all of the available data wherever the patients are, because we have a relapse endpoint, and the relapse could happen long before 72 weeks. So they get all of the relapse events are really the most important driving force behind the sample size of the here, and that's what they will check on an unblinded basis.
Is it, the relapses have been going down. Look at event rate separation. We need events to separate.
Yes. So I think event rates or relapse rates are very important. Yes, we learned this last year from a phase 3 outcome there as well, that sometimes event rates is quite important. We monitor this on a blinded basis, of course, and we're very much convinced that we're on track. However, you can only really assess this on an unblinded basis eventually. Yes, because then I think you have to see certain amplitude of effect as well. But this is something we cannot be involved in. This is purely done by the IDMC, and they will give us recommendations. But it ensures, I think, that what we believe on a blinded basis, that we are on the right track in terms of relapse events, that is confirmed by the IDMC, which I think gives us confidence that we're continuing on the right path with this trial. Good.
Good. Thank you, Andreas. And I like that we have the chance to really present some more deeper insights than usual. If you just give the company presentation, you always stop at the top of things. And as you may see that here in this case in multiple sclerosis, I think the medical need is so closely correlated with the progress of science that I think that makes it a pretty exciting place to be, and put us a bit of pressure on us to deliver something now, because we, after the recent results from this one BTK study and potential with other BTKs to follow, there's not too much in the pipeline right now to help those patients with something which could go beyond the just normal anti-inflammatory treatments and targeting the relapses. But now I think what is also important, if we now progress towards the finish line, how can we secure the IP? How can we make sure that we get the exclusivity for the drug? And from the very beginning, we started to build a very broad, multi-layered portfolio of patents and also other IP around the asset, and wanted to make sure that we cover all the different ways to protect Vidofludimus calcium from generic competition. And where we are right now, and I added some more slides this time here to disclose a little bit more where we stand, because last year we published 2 patents being granted by the USPTO for the drug, and another one we published just recently. So we really, you see that the company is active in broadening the protection. And the intellectual background here is really to, on the one hand, protect the compound and the material we use. And this is, on the one hand, the calcium salt on its own, which would protect, it's a granted patent, and duration is into the 30s, and with enhancing the exclusivity with the patent term extension, PTE, could protect us until 36. But what we believe is we bring together here with all the different layers is that altogether, at least until 41, we believe we can expect exclusivity. On top of the compound, and part of the compound is also the polymorph we are using in our tablets we are currently using in the phase 3 and the phase 2 studies, is the dosing and the strength of the use, specifically in those indications. We have a dosing regimen patent, which was granted last year in the US, and another patent of the dose strength now in RMS, and we also want to broaden that into progressive MS. And those patents also can bring us into 41. And on top of all of that, also we, this is a new chemical entity, so we will also be subject to regulatory exclusivity on top of the patents. But we didn't stop there. We had more things ongoing. So, for example, the indication. So on the one hand, we have the RMS protected via the dose strength patent, which was granted last year. And we also applied for in a division application for progressive MS here for the dose strength patent. And what is a pretty exciting new patent we filed, and we didn't disclose something specific here on that because it's not yet published, is of course treating activating of Nurr1, and that by that potentially treating PIRA could also even bring us beyond 41 as exclusivity. For example, this patent would be 44. On top of that, we also have some backup molecules. And one example here is the deuterated version of Vidofludimus, the new compound patent, which is running into until 42, and considering PTE would bring us exclusivity to 47. But considering the maximum you can get is 14 years of exclusivity. So all in all, message is here, we have a very broad portfolio of multi-layered patents protecting the asset. So now where are we and what are we planning to do? And I think I don't want to repeat too often what was already said today. So we have these studies ongoing. There's a lot of excitement now about both studies, ENSURE program in the phase 3, but also the phase 2 CALLIPER study and the data readout next April. And we think that this molecule can change with these data, can change the way we treat multiple sclerosis in general. Here's a look on the timelines we spoke about. So ENSURE futility analysis late this year, and then April 25, we expect the PMS CALLIPER readout, and then in 26, the ENSURE readout. So how is that positioned in the world of current treatments for multiple sclerosis? And it's true, it's a huge market. So these are numbers from 22, where the leading brands together had a turnover of 23 billion of revenue. And every single molecule on that list still was a blockbuster molecule, despite some of those being very old treatments. So that's definitely an area of importance and commercially relevant markets. And this is a slide I've shown you before with a little bit of slide modification. So these are the first two waves I mentioned, and they make more or less these 23 billion revenue. So the question is, what is the missing piece? And I hope we could convince you today that the missing piece is something we have together now with Vidofludimus calcium, which is on the one hand, having a safe treatment for MS patients, more following the route of traditional treatments, but on top of that, offering a potential neuroprotection. And what happened, and it's also something which a little bit of serendipity and happened in the last year, was that there was a big group of compounds developed by several pharma companies on BTK, which have been very promising initially, and most of the drugs have a clinical hold, at least in the US, because of liver toxicity issues. And then end of last year, there was one, the Immunic study read out and didn't deliver any benefit over their comparator, which led to discontinuation of that further development. That, as I said, that makes Vidofludimus calcium more, it's more exposing Vidofludimus calcium to patients and the market and other stakeholders, because it's one of the rare remaining options we have in development, which could serve as the first, for example, first treatment for non-active secondary progressive MS, and also give the benefit of neuroprotection to patients with other forms of MS. And coming back to the initial slide I've shown you as a third slide in the presentation, we want to fill that piece here to, on the one hand, address RAW, so relapse-associated worsening, and PIRA in one molecule, and doing that by having a drug which is absolutely well tolerated and has a very attractive safety profile. Putting that a little bit in the context of segmentation of the market and patients groups, we think on the one hand, it's important for patients to have a safer treatment option, and to also offer something new for patients who need alternative on their current treatments, because they start with something and they either don't tolerate it or they don't experience enough protection, and therefore having a more early treatment option which offers a unique safety and tolerability profile, which is by itself a big market opportunity. On top of that, I think confirming neuroprotection would extend that substantially also on the volume and pricing side to address disability progression and also other aspects of disease like fatigue. And maybe for the first time, have an option, treatment option for patients who are absolutely not treated because of their little or not willingness to tolerate safety and tolerability issues. So where do we stand and what's coming up? So we already achieved in several clinical trials a very strong clinical signal for the drug, that the drug works. In the emphasis phase 2 study, as Andreas has shown, we have seen the interim data on NFL in the phase 2 CALLIPER study. And now the next big milestone is the interim readout of the phase 2. The next milestone will be the fertility analysis and the ENSURE phase 3 program. And very importantly, in the year from now, we are urgently waiting for getting data from the CALLIPER PMS study. You've seen that slide before because I like that the graphic here, and as Andreas said, we think that Vidofludimus calcium is standing out of the crowd. It's the blue balloon on the slide here because it combines 3 important features to serve as the future treatment for multiple sclerosis, including relapsing and progressive MS, neuroprotection, anti-inflammatory effects, and antiviral effects, combined with an outstanding safety and tolerability profile. Good. And with all of that, we believe that Vidofludimus calcium is targeted to elevate the standard of care with a overall holistic solution for a full spectrum of MS patients. And with this slide, I want to conclude the presentation and happy to answer questions on this and every part of today's presentations.
One question for the standing ovation, but what's the registration strategy for progressive MS?
So what?
What's the phase 3 registration strategy for progressive MS?
Once again, I think maybe Andreas can support that. It's based on the data because it really depends what comes out. It may be there is an accelerated or expedited way to get it approved. Because the study is not small, it could be that the subset of non-inflammatory SPMS patients is big enough to demonstrate the effect size on a clinically relevant parameter. Or it needs another phase 3 study, but definitely we believe that we don't need 2 studies. And also that was more or less a conclusion from our interactions with the FDA that we, in case we would need a phase 3, only one would be enough for this.
The precedent is the CD20 primary progressive.
Yeah, it was one study. And it was not super big. It was something which is manageable.
Yeah, we had one more question here, which I think, Daniel, you answered already, but maybe the second part more for Andreas. How do you view the use and positioning of Vido in MS patients, and what percentage of MS patients do you expect to be treated with? Now I need to hand you.
I'm not sure that I can answer that question.
Me too. I think at the end, everybody would deserve to receive Vidofludimus. The question is really, what is the differentiating factor? I think if I would be a patient and look on that, there are these 2 sides. One is the safety and tolerability, which if you're a young patient, it's a very different answer than if you're maybe 65, 70-year-old. So I think the data we have generated so far would speak for both, but difficult to really make a quantitative assessment.
Thank you. Any more questions here in the room? No? All right, then I would say we conclude. Now it's time for the standing ovations. Thank you so much for listening, for attending here in person, and also for attending on the internet. Thank you. And if you have any follow-up questions, let us know. Of course, we are still here or available via email and happy to answer any questions. Thank you.